And at the worst possible time, my slide presentation fails. Okay, on behalf of myself and my co-authors, I extend thanks and greetings to participants and organizers of Evolution 2025. My lead author, Bill Basner, is an emeritus professor of mathematics. John Sanford is a retired Cornell research professor and genetic engineer, famously known for his invention, the gene gun, which, by the way, is in the Smithsonian National Museum of American History. Ola Hasjer is a professor of population genetics at Stockholm, University of Stockholm. I'm Salvador Cordova. I'm presently a PhD student in biomolecular engineering at Liberty University with 5 previous science degrees, including a master's degree in applied physics from Johns Hopkins University. I was previously a senior engineer and scientist in the aerospace and defense industry working for MITRE before getting retrained in biology by John Sanford and studying at the FAES Graduate School at the NIH. We report on ongoing work that builds upon our previously published works in population genetics, protein biology, structural bioinformatics, and unpublished works in biophysics. So what brings me here today? It is this comment by Richard Lewontin, who said, the problem is that it is not entirely clear what fitness is. And no concept in evolutionary biology has been more confusing. Further, he laments the incommensurability of the original Darwinian conception of fitness with the definition of fitness in modern population genetics. My goal here today is to add even more confusion to the issue and incommensurability. Just kidding. Apologies in advance that I have to blast through these slides. Lewontin's lamentations are echoed to varying degrees. I list some here that can be investigated by further rewatching recording of this talk in slow motion. Sorry, but we presenters are all under tight time constraints today. I recently queried Google Generative AI and asked, why is evolutionary fitness ill-defined? It responded, evolutionary fitness is an ill-defined term because it's a complex, context-dependent concept that's difficult to measure and define precisely. Charles Darwin wrote of endless forms most beautiful and most wonderful, organs of extreme perfection and complication. Yet paradoxically he said, what a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature. For Darwin, the process of natural selection resolved this paradox. Darwin himself wrote, the presence of much suffering agrees well with the view that all organic beings have been developed through variation and natural selection. That said, it has become fashionable in light of recent discoveries in biophysics to say life is more perfect than we imagined, which is also the title of a frequent and popular lecture by William Bialik, a Princeton biophysicist and NAS member. It is easy to believe biological systems are technologically inferior to human-engineered systems for various reasons. First, there is, as Darwin pointed out, lots of suffering, blundering, cruelty, and waste in the world. Second, human-engineered systems often outperform biological systems in certain, but not all, metrics. Third, a far more subtle but significant issue is something illustrated by Shannon's noisy channel coding theorem. That is, communication systems that are optimized for compactness must, as a matter of principle, allow large amounts of errors to enter a communication and/or storage system that are later remediated by error correction systems such as Reed-Solomon and Bose-Chuduri-Hakogen schemes. By way of comparison, DNA has numerous ongoing breakdowns and errors, but DNA repair mechanisms correct almost all of those errors, and DNA is one of the most compact storage representation there exists on the planet. So, as a rule of thumb, the more complex, compact, and high-performing a system is, the more prone to breakdown and errors it will be. Hence, we can be misled to think an otherwise high-tech device is deeply flawed. With that in mind, here is one example of a feature in biology that was previously viewed as clumsy and blundering, but then later termed as ingeniously designed. The phrase ingeniously designed was used in a PNAS paper addressing specifically the backward wiring of the human retina. This is a case example of something previously viewed as clumsy and blundering, only to later be deemed ingeniously designed. As a former engineer, I love that phrase, ingeniously designed. Fasten your seatbelts. In the interest of time, I now have to go into machine gun mode and fire off a long list of examples, which should be easy to track down by replaying a recording of this talk in slow motion. Life is more perfect than we imagined, illustrating fitness through physics. Some superior technological secrets have come to light from a deep-sea organism. Modern technology cannot yet compete with some of the sophisticated optical systems processed by biological organisms. Next, bird quantum magnetic compass. Bird quantum magnetic compass operates at the quantum limit. Next, exciton transport is 100% efficient in photosynthesis, which human engineering can't ever exceed in solar cells. Next, Limosa laponica can fly 9 days straight with no stops to rest or sleep. 11,680 kilometers, weighs 10 ounces. We can't make anything today so energy efficient. Next, the human brain operates at around 25 watts. A comparable silicon-based machine would require 10 million watts. The brain operates at the extreme limit of thermodynamic efficiency defined by the Landauer principle. Therefore, human designs will never do better. Next, enzymes are biocatalysts that operate with catalytic efficiency better than anything we can build from scratch, especially enzymes like topoisomerase, which I happen to have published on. Sharks can sense electric fields generated in the muscles of its prey at the limit of what is physically possible. The eye can detect a single photon that knows single molecules. Therefore, human designs cannot do better as a matter of principle. Natalie Angier, after interviewing several biophysicists, said that biological systems couldn't get faster, more sensitive, or more efficient without first relocating to an alternate universe with alternate physical constants. Emmanuel Todorov at University of Washington mused, "You might say, well, the human body is sloppy, but no, we're better designed than any robot." This also agrees with the work of Stuart Burgess, professor of biomechanics and robotics for 40 years, who incorporated award-winning biologically inspired designs into robots and spacecrafts. And the final frontier of excellence in biological systems is in the area of self-assembly, self-healing, and von Neumann self-replicating automata, areas in which human engineers cannot even begin to compete. I worked in biomolecular systems, and self-healing is just a nasty problem we haven't solved, but it's solved in biology. Nobel Prize winner Sidney Brenner said von Neumann's automata should be central to biological theory. The best example of Turing and von Neumann's machines are to be found in biology. Nowhere else are there such complicated systems. Code script must form the kernel of biological theory. Pause. How do biophysical benchmarks, as just illustrated, relate to the notions of fitness and adaptation? This is really the central question of my presentation today. These physical benchmarks define the maximum possible level of fitness and adaptation in the engineering sense. The conflict between the prevailing definition of fitness in population genetics versus the engineering definition of fitness from physics came to a head when I worked on the problem of fitness maximization, especially while studying Grodwall's extraordinarily compelling paper, "The Theory Was Beautiful Indeed, Rise, Fall, and Circulation of Maximizing Methods in Population Genetics." Nowhere in my reading of this historical magnum opus was there any mention of biophysical benchmarks used in determining maximum possible fitness. Yet obviously the bird quantum magnetic compass has maximized its performance, dare I say fitness, in magnetic sensing. Same for excitons and photosynthesis, energy efficiency in the human brain, etc. So how do engineers determine if something is adapted to fly? If one asked an engineer how a bird or butterfly is adapted to fly, he would say it is adapted to fly because its parts and organizations are consistent with engineering principles of flight. Basically, for something to be adapted to fly, its wings must be able to generate enough lift to overcome the force of gravity. Its engines must create enough power to enable the airplane to overcome the drag caused by the air, so its wings can generate enough lift, etc. That illustrates the engineering view of the meaning of adaptation, and unfortunately it doesn't at all involve adaptation by appealing to reproductive efficiency. From numerous experiments, it now appears the engineering viewpoint of fitness is anti-correlated with the population genetic viewpoint of fitness. These experiments serve as counterexamples to the ultra-Darwinian viewpoint as expressed in popular books such as the blind watchmaker and Darwin's dangerous idea. Ironically, Darwin's dangerous idea advocated the view that biology is engineering, and if we want to get more engineers involved, this is the way it's going to have to go, but they have lots of money. If I may point out an important aside, Wolf and Kuhnen wrote, quantitatively, the evolution of genomes appears to be dominated by reduction and simplification, punctuated by episodes of complexification. Punctuated complexification. I like that idea, but unfortunately, it may not be subject to repeatable experiments. That said, the idea of rapid punctuated complexification may be supported by the idea that proteins are instantiations of Platonic forms. Thus, there may be no gradualistic Darwinian route to evolving some of these proteins. When I look at amino acid residues of collagen on the left and human zinc finger 136 on the right, and colorize the residues, it suggests to me there is no common ancestry from a single ancestral gene locus for all proteins, nor a gradual stepwise route for their emergence. This problem would seem even more acute for proteins whose function is critically dependent on multimeric quaternary structure, like topoisomerase, thus reinforcing the notion that proteins instantiate Platonic forms. Additionally, consider this experiment by von Hofwegen et alia, which showed that the DCSU protein in long-term evolution experiment never recovered function over 80,000 generations following a mere 5 nucleotide deletion event. This experiment supports the anti-correlation hypothesis, but I would hope for hundreds more of such experiments. In some cases of reductive evolution, it may be powered by natural selection, as in this study, again reinforcing the anti-correlation hypothesis. Even more pointedly, the famous long-term evolution experiment shows anti-correlation between engineering fitness and population genetic fitness. Cooper and Lenski showed fitness increased in one environment and declined in multiple other environments that tested catabolic efficiency. And this is strong evidence in favor of the anti-correlation hypothesis. And beyond the fact we are now in the 6th great extinction, the biosphere may be in an era of reduction and simplification, to use Wolf and Kuhnen's words. And declining genetic health over time is a testable prediction, at least in principle, that could lend support to the anti-correlation hypothesis between the engineering perspective of fitness versus the population genetic definition of fitness. But in the meantime, let us all work together to preserve and study the ingenious designs in nature composed of organs of extreme perfection and complications that constitute endless forms most beautiful and most wonderful. Thank you. Questions?
Any questions from the audience or in the Q&A? There are some questions in the chat. I don't see something on the Q&A right now, but in the chat there is a question. Do you have a list of the publications you mentioned from? And then there is another question. Would you like to first address the first question? We still have
yes.
a minute.
Oh, certainly. Thank you for the question. And unfortunately, the best list is the one actually in the video, and one would have to watch it in slow motion, but I should provide that. I think my email address is in the attendee directory, and please write to me and communicate, and I would be happy to try to type that up for you. Thank you so much for your interest.
Okay, I think it's time to move to the next speaker. So Shen is our next speaker. Thank you, Sal. How you can share your screen when you're ready.
Can you see the screen?
Yeah, that's fantastic. Thank you. When you're ready, let me know when I'll start the timer.
And you can see the slides, right?
Mm-hmm.
Okay.
Perfect. Okay.
Shall we start?
Yes, thank you.
Okay. Hello, everyone. I am Shen in November Lab from University of Chicago. Today, I will present fine resolution asymmetric migration estimation. Asymmetric migration on gene flow is a common phenomenon in nature. It can be driven by directional forces like the prevailing winds or like ocean currents. Can also be due to the historical or demographical factors like human range expansion. However, detecting these asymmetric gene flow from the inference is difficult. An important trade-off exists in current inference tools. So we either assume the migration is symmetric to get a finer resolution, which means that the number of demes, more number of demes, like is done in EAMS and FEAMS, or we allow the migration to be asymmetric, but restrict ourselves to coarse resolution, which means just a very small number of demes, like is done by Lundgren and Ralph in 2019. The reason that such trade-off exists is that these like these methods are based on common inference procedures. So all of the both methods uses the structure coalescence. So from the lineage migration rates or the backward migration rates and the coalescence rates, we can compute the expected pairwise coalescence times. And from which we can, the expected pairwise coalescence time will influence the variance structure of the samples. However, computing this expected pairwise coalescence time from this backward migration rates and coalescence rates is not an easy task. And the trade-off I present before in the last slide actually shows different approaches to solve this computational problem. So here's the equation for pairwise coalescence times. Here D is the Deme number. Gamma is a D by 1 vector of within Deme coalescence rates. T is a D by D matrix of expected pairwise coalescence time across Demes. And L is the graph Laplacian or the negative transition rate matrix constructed from backward migration rates. So we can see that this equation, what we want to do is we want to solve this T, the matrix of expected pairwise coalescence time, given the knowledge of the coalescence rates represented by gamma and migration rates represented by L. So there are the order of D square variables, and it's a linear equation. So if we directly vectorize this equation and solve it, the complex, like it's done in Lundgren and Roth 2019, then the complexity is actually D to the 6. And if we want to, in a lot of cases, it's basically prohibitive. On the other hand, if we use the symmetric assumption and other approximating strategies, like is done in EAMS and FEAMS, we can reduce the complexity to order of D cubed. However, we have a very strong limitation. We limit ourselves to the symmetric assumption. So the question is whether we can combine the strengths of both methods and develop an exact method to solve this equation with acceptable computational complexity. Okay, so we figure out how to solve this class of generalized Lyapunov equation exactly with time a complexity of order of D4 by writing the solution as a linear combination of order of D Lyapunov equations. Each can be solved with order of D cubed. Combining this technique with the panelized maximum likelihood estimate approach, we develop a new method called FRAME, which stands for fine resolution asymmetric migration estimation. So the right-hand side figure shows a comparison of FRAME to the brute force method by directly vectorizing this equation, and we can see that the FRAME is actually much faster than the brute force method. We test our method using simulations. Here we would like to show the simulation results as well as the basic representations of FRAME. So this is a ground truth topology for lineage migration we designed to test the performance. So on the left-hand side, you see the full graph. So basically, we draw each edge, and the strength of the migration is shown using the color. If it's blue, it means strong migration. If it's yellow, it means weak migration. On the right-hand side, we see a graph we call the difference graph. So here, instead of draw all graphs, so if we have a migration from A to B with migration rates larger than the migration from B to A, then we will only draw an edge from A to B. representing the log, the difference of the log migration rates. So here the color represents the direction of the migration, and the strength is a stress of asymmetry is shown using the transparency. So a topology like this here is what we call a large-scale pattern of spatially converging lineages, because we see lineages converging to the center in the difference graph. So we test our method by first doing a dense sampling, which means that we sample all the DIMs, and in each DIM we sample 10 individuals and 100,000 SNPs. And we can see that for the topology of large-scale spatially converging lineages, frame nicely recovers migration pattern. We then test our method's power for sparse sampling, which means that you only sample 25% of the DIMs, and still other settings remain the same. And in this case, we are still able to roughly recover the migration pattern, especially when you look at the difference graph. Similar results hold for other large-scale migration patterns. For example, in contrast to previous example, this is the ground truth topology for spatially diverging lineages, where lineage diverge from the central point. And we also recover the patterns nicely in the case of dense sampling, and roughly detect the pattern in sparse sampling, although it's not as good as the case of spatially convergent lineages, but we can still see that, especially from the difference graph. So here is another example for large-scale patterns. This one is simply the ground truth pattern of large-scale directionally migrating lineages migrating from the left boundary to the right boundary. And recover it in the dense sampling case, and roughly recover it in the sparse sampling case. The things becomes more interesting when we go from large-scale patterns to small-scale patterns. This is a showcasing example of a ground truth topology with a wrapper of small-scale patterns. You have spatially converging lineages, spatial diverging lineages, you have this directionally migrating lineages, and you have cycles. So we are able to recover this pattern in the dense sampling, even if this is a very complicated pattern. So in the sparse sampling, things become more interesting, because in this case, the patterns are only partially recovered when the sampling is sparse, and it works better if you have more samples around the pattern, and worse if you have very few samples around the pattern, because this pattern is small. So all the above simulations are assumed to be in the migration drift equilibrium, which is what frame design is designed for. However, we also test cases where this assumption is violated. So here is an example of a range expansion. We also sample all DIMs and the same setting. Range expansion, so forward in time, we see a range expansion from the central line to both boundaries. And backward in time, we'll see lineages migrating from the boundaries to the central line. And we see that frame is able to detect the signal. So although this is not like, we don't know what is the ground truth because it's not an equilibrium case, but we do detect the signal of this range expansion from both like the foregraph and the difference graph. The frame can detect these signals even in this highly non-equilibrium case. So we also apply our method to some empirical data sets and find that the results vary across species. For example, in the North American gray wolf dataset with 180 individuals and 80,000 SNPs, we compare our results to previous method themes. So if you look at the R-square of the fitted genetic distances versus empirical genetic distances, the themes, we have R-square equal to 0.964, and frame slightly increase it to 0.97, and showing that the symmetric assumption made in themes is rather reasonable. On the other hand, we look at the populace data set, which has 431 individuals with 31,000 SNPs. And this is also used by Lundgren and Rolf in 2019. And in this case, things becomes very different. In themes, the R square is only 0.869, and in frame, it's 0.99. Frame significantly increase R square. And we also see very clear pattern of asymmetry. We see the directional migrating lineages at the top of this different graph to the northwest. And this coincides with the findings in the original research. So in this data set, it might be inappropriate to ignore asymmetries. Apart from using current genetic data, we also apply our method to ancient DNA data. So this is a human ancient DNA data from 3,500 BC to 1,500 BC Europe. It has 1,361 individuals with 51,000 SNPs, and these are like filtered from the Allen ancient DNA resource. So this is known to be the period of the expansion of the steppe expansion. So the Yamnaya population expand towards both east and west directions. So in our fitting, we also find very clearly such signals as shown in the green box. Because it's a backward migration, so you should like understood these directions in the reverse way. So basically lineage move backward to this center. And this is an example because we know human migration is highly non-equilibrium, and it is consistent with our simulations. In the future, we think we can further the research in two directions. One is we can directly model the non-equilibrium situation instead of fitting the non-equilibrium cases with an equilibrium model. Second, instead of using inference method based on a variance structure of the samples, we can use IBD-based method, such as using the length distribution of IBD segments, like is done in MAPS. Thank you.
Great talk, Hal. Thank you very much. Are there any questions from the audience? You can raise your hand, or you can write in the Q&A, anything that works. No questions from any of the participants. I do have a quick question, Hal. What would be the next steps for verifying these?
Next step, you mean?
Mm-hmm.
You mean these two? Which one?
Yeah,
You mean the non-equilibrium one or the IBD?
the IBD. Sorry.
Because maps use the length distribution of IBD segments, and a key difficulty in maps is to solve an equation that is similar to what we have just shown, and we can apply similar technologies there, which means that in principle we can develop an asymmetric version of maps.
Okay, that sounds good. Great, thank you. I think we can move on to the next speaker. Miles, you're up. Thank you again, Hal, for your talk.
All right. How does that look to folks? Cool. Should I go ahead and get started, or should I wait till 2? Okay, I'll get started. So hello, everyone. Really happy to be here. Thank you to the Society for hosting this meeting. Today, I'm going to be talking about a pair of papers that I wrote recently with some collaborators and my advisor, Emily Josephs at Michigan State University, where I recently graduated from my PhD program. So happy to hear any thoughts, questions y'all have on this work. I think I'm in the PopGen theory section of this conference because the motivating driver behind this research goes back to neutral theory. And I imagine this audience is pretty familiar with neutral theory and what it is, but just in case, I'll take a moment to explain it. It's this sort of big idea in population genetics, right? Where we expect a lot of the genetic variation we observe in populations to be selectively neutral, because variation that's acted upon by fitness could, like beneficial variance, could quickly rise to fixation, and deleterious variance would be quickly eliminated, leaving mostly neutral variation. So to kind of represent that with more of a picture, we can imagine the amount of genetic variation in our population as sort of the level of water in a pool. And in this analogy, then, the process of mutation is sort of like the tap, the faucet that is adding more variance, more genetic variation to our pool. And the drain on our pool is genetic drift. So here again, we're assuming that the action of selection on genetic variation and other factors is relatively minor. To represent this with an equation, it's often represented this way with theta equals for NE mu, where theta represents the genetic variation, so the level of water in our pool. NE is effective population size, which is where drift comes into play, and mu is mutation rate. Now we know that from big meta-analyses that mutation rates tend to vary within the same order of magnitude across many species, whereas we know that population sizes vary across many orders of magnitude. So neutral theory gives us this nice testable prediction that we expect larger populations to have more genetic diversity, which is a pretty simple idea. But famously, when we go to test that prediction in real data, it doesn't always match up. And this is a nice plot from Vince Buffalo that I think demonstrates this nicely, where each point here is a different species, and they're plotted according to an estimate of how many individuals are in that species versus their level of genetic diversity. Now, if that equation that I showed on the previous slide was a good model for nature, then most of these points should be falling within this gray shaded region right here. But as you can see, most of the points here fall way below their neutral theory expectations. And so this gap between theory and expectation is often referred to as Lewontin's paradox of variation, because Richard Lewontin wrote about it in 1974, and we've been studying it for the past 50 years since. Now, there's a lot of really cool biological mechanisms that contribute to Lewontin's paradox. This is a really awesome review by Charlesworth and Jensen and GBE. I recommend you read if this is of interest, and goes over lots of different biological mechanisms that contribute to Lewontin's paradox. Today, I'm not going to talk about any biological mechanisms per se, more so ask the question, are there any technical mechanisms, technical factors that also contribute to Lewontin's paradox in addition to these biological mechanisms? In other words, could some of this pattern be potentially explained by just underestimating genetic diversity in many species? And why do I think that is? One reason is because pangenomic analyses, where you have more than one reference genome for a population, have really expanded in recent years and have shown us that our sort of standard approach of measuring diversity, where we have one reference genome and take population reads and align that to our reference to estimate diversity, it's often a really good approach, but it has some problems they're collectively called reference bias. So what do I mean by that? I'll give you some concrete examples. So you can imagine this blue rectangle here as our reference genome, and these green rectangles are population reads that we've sampled, and we're aligning it to our reference to identify SNPs and calculate diversity. Now, it's pretty common in these analyses to end up with some proportion of reads that do not align to your reference. And it's possible some of those reads are contaminants, but it's also possible that these leftover reads represent real genome sequences in your target species, but that region is just not represented in your reference. And so you're not able to look at variation in those regions. Another way reference bias can come up is maybe the target region does exist in the genomes of your species, but it's maybe too far diverged from the reference to align accurately. So here I've added some yellow boxes to represent mutations, and if enough of these accumulate in a small region, it's hard for alignment to accurately assign them to your reference genome. Now, there's lots of pangenome studies that demonstrate this phenomenon where if you have just one single reference genome for your species, you actually miss out on a lot of really interesting SNPs and structural variants and other types of genetic variation that are out there. And here are just a few examples. So sort of the key question here we want to ask is, does this sort of reference-based approach, this single reference-based approach, also contribute to Lewontin's paradox in some way, perhaps by causing us to underestimate genetic diversity? And I'll talk a little bit about how we did that. First thing was to assemble a data set and measure genetic diversity in two different ways. The first way is the sort of standard reference-based way that I just talked about, where we have some reference sequence, which is maybe our genome sequence here, and then we take some population sequences, align it to our reference, find single nucleotide polymorphisms, and do our standard calculations of genetic diversity. The second way, we could estimate genetic diversity. It doesn't rely on reference genomes at all, and instead uses a tool called K-mers. And K-mers are small subsequences of larger sequences. So, for example, here I could take this purple sequence and decompose it into subsequences. I could say the first one is GCAAC, and then I add that to my list over here. Then I shift my window over one base, and the next group of 5 is CAACG. Add that to your list. Repeat that for all of your reads, all of your samples, and you end up with these lists of K-mers for each sample. And you can tell just at a glance that these lists of K-mers look different, and that's because of the presence of this SNP here in our original diagram. But these K-mer lists, as you imagine, could also change in response to other types of variation, structural variation, presence-absence variation, and so forth. But it doesn't rely on alignment to a reference genome at all. Here, just a quick note, I'm showing the concept here with K-mers, just with K-mers of length 5 for clarity, but in the real study, we use K-mers of length 30, so quite a bit longer. And then the way you go from these K-mers to an actual number representing a genetic diversity measure is you take samples, one pair of samples at a time, and you do basically a set operation where you look at the ratio of how many K-mers are shared between the two samples versus how many K-mers are exclusive between them. And the degree of overlap between these two circles tells you how genetically different the two samples are. And you sum that across all your pairs. So a sort of baseline question we want to ask ourselves first, right, is how does this K-mer-based approach of measuring genetic diversity relate to the more standard approach, usually called nucleotide diversity, where we focus just on SNPs from read alignments? And I worked with two amazing collaborators, Olivia and Robert, to look at this question, where we simulated a bunch of populations focusing on just single nucleotide polymorphisms and assuming that there's no reference bias. Our reference genomes are perfect. This is the sort of relationship we find. So here, each one of these dots represents a simulated population, and we're plotting them according to sort of the standard metric of diversity based on SNPs versus these K-mer-based metrics I'm just talking about. And the different colors represent different lengths of K you could use to look at K-mers. And in each case, the correlation between K-mer diversity and SNP diversity is about 0.97. That's the R squared. While the exact slope of the relationship will change depending on what value of K you choose. If you use the same K to compare two samples, the R squared is 0.97. So what this tells us is that if we measure genetic diversity in these two different ways, and we're just looking at, and in the simple world where only SNPs matter and there's not a reference bias, then these two different measures should tell us the same story. So do they actually tell us the same story in real data? So we next got together this really large data set of publicly available genome sequencing data, about 100 species and 200 trillion bases of DNA sequencing data. These are all plant species, and we reanalyzed all this publicly available data with the same bioinformatic pipeline, which took about a year. And that was to try to limit the influence of bioinformatic bias if we were to just take these estimates from previous studies, which may be each analyze their data slightly differently. This is a species tree here of all the different plant species we included. And these bars here are the sort of primary variables that we're interested in. So this pair is standard measure of diversity versus a K-mer metric. And this group of blue bars here is the some population size proxies. And I'm not going to talk about this, but I'm happy to answer questions about it. But the second variable we need to know is estimates of population size for each of these species, and you have to rely on proxies to do that. And so we use range size and body size as our two proxies of population size. So at the last minute or so that I have, just want to quickly, go back and test this hypothesis and see if these K-mer-based and SNP-based measures tell us the same story. So what I'm going to show you now is, again, each of these points represents a different species in our data set, and I plotted according to these population size proxies I talked about. Happy to answer questions about that, versus a more traditional way of measuring genetic diversity, so relying on just SNPs identified from mapping reads against one reference. And what we see is that this relationship is pretty flat in this data set, which kind of mirrors Lewontin's paradox, right? We expect there to be a positive relationship between these two variables, but when we actually go to measure that, it ends up looking pretty flat. So now this is the same relationship, but just looking at K-mers instead. So again, the horizontal axis here is the same, population size proxy, but now we're just looking at a K-mer-based diversity measure instead of a SNP-based diversity measure. And when we do this, the relationship ends up being a lot steeper and being statistically significant. And we repeated this analysis in many different ways. We tried different measures, different calculations of K-mer diversity in different types of population size proxies, and they all showed the same answer. So kind of going back to how I started this talk, I think that this suggests that that also, in addition to these very important and very cool biological mechanisms, there's some component of this paradox that could just be due to how we measure genetic diversity, right? And I think pangenomes can help us elucidate that a little better, because there are kind of two big caveats I want to leave you with that I think are kind of driving the next steps of what I want to do. And that is, one, we don't know how important this underestimation of diversity is relative to other factors in explaining the paradox, right? And the second one is, we don't know what concrete variants are driving this trend. We know that K-mers have a different relationship to SNPs, but we don't know the exact SNPs or whether structural variants are what the K-mers are capturing to drive this trend. But I do think that now a couple of years have passed, we have a lot more pangenomes, and we could look at this question with a lot more precision. I think would be really interesting. So thank you all for listening. A lot of people supported this work, thanks to the NIH and NSF. And these are the two papers that I talked about. Happy to answer any questions.
Great talk. Thank you very much, Miles. Are there any questions from the audience? You can always raise your hands or write in the Q&A. I have a question while we're waiting for a question from the audience, because you did briefly mention structural variants, and I was thinking that we have the issue of the duplication, that a lot of the duplications, they're direct duplicates, right? So in that case, you could expect to have gene conversion, which will neutralize the background. So overall, the estimation will be an underestimation of the variance, which the variance was present, but then is masked. And this is one aspect of it. And then the other aspect is how the different lengths of the K-mers or the choice of the K-mer length can influence this, and if you've tested it with different lengths.
Yeah. So I'll start with the second part there. The choice of K. So yeah, so in the empirical data set, we didn't try varying the length of K in that study I mentioned, mainly because each time you repeat the analysis for the whole data set, because it's pretty large, it would take an extra year of computation time to repeat it for another length of K. But the good thing is that K-mers in the range of 30 tend to be very well used in plant K-mer-based analyses mathematically. And once you get up to K in the range of about 30, it's highly unlikely that two samples will share a K-mer just due to random chance alone. And so that helps it be a much better estimate of how genetically dissimilar two samples are. So yeah, that's the short answer. We did use the same length of K for every for every species and every individual to keep that the same. And this paper on the left here kind of shows mathematically why that tends to work across a pretty large range of genome sizes. In Arabidopsis, about 97% of 30mers map to unique locations, which yeah. And then
I'll have to stop you here,
Okay.
but you're absolutely welcome to continue the conversation in the chat. We will have to move on to the next speaker, that is Zarus. Sorry for interrupting.
all good.
Tight schedule. But thank you very much. Great talk. I think you're muted. Would you mind?
There we go. Sorted.
There, thank you.
All right.
Sorry
So
if I mispronounced your name. We are ready to start when you are. Just share your slides with us and we're good to go.
all right, can you see my screen?
No.
What? Hang on. It's working before. There we go. Can you see my screen now?
Yes.
Excellent. All right. So my name's Zarus, and my presentation is about the dispersal of modern humans across Africa. I want to infer if this dispersal, if this expansion happened, and where it originated. Overall, I find evidence of a Southern African origin for this dispersal, but I discuss reasons why there are reasons that this finding is very questionable, shall we say. So modern humans originated in Africa, either with a single origin, or we originated across the continent of a Pan-African origin. At some point, we globally dispersed out of Africa. There are a lot of unanswered questions about what happened in between our origin and our global dispersal. Was there an across Africa expansion? If this expansion happened, did it go into unpopulated regions, or did it go into populated regions with admixture occurring or not occurring? So in addition to asking, was there an expansion, we also want to find out where it originated from. The most popular candidates for the origin are Eastern Africa and Southern Africa. And as I point out here, the origin of the expansion need not be the origin of modern humans. It's not the same question, always. All right, so we can use the effects of population bottlenecks to infer if an expansion occurred and where it originated, because population bottlenecks cause genetic drift to increase, which causes diversity to decrease the further away we move from the origin, and it causes linkage disequilibrium to increase. So overall, when looking across the literature, it's unclear if diversity and linkage disequilibrium have patterns which are consistent with an across Africa expansion. In a study by Brenner-Hen and colleagues, a southern origin was indicated through linkage disequilibrium. Karina Schlebusch and colleagues, they used various genetic variables, but did not find that a regional origin was indicated in particular. For these variables, using only about 20 populations at a time, they showed populations values on maps of Africa, and that seems to have been their way of gauging if an origin is indicated. Is that enough? They do not mention testing if there were statistically significant trends within Africa, and they do not seem to have used a sufficiently objective way of assessing where the origin seems to be. Interestingly, Hunley and Cabana used Eastern Africa as the origin in their analyses, and they did not find a trend in autosomal diversity. But in a preprint a few years back, I found that autosomal diversity fell as distance from Southern Africa increased. So what I wanted to do was to get an objective general impression about the expansion. Did it happen? Where did it originate? Using different variables and different data sets. So how did I do this? I examined cranial and genetic diversities in sub-Saharan Africans. I calculated cranial shape diversity using two data sets, the Howell data set and the Manissa et al. Data set, which is a subset of the larger Hanihara data set. Also used some genetic diversities, which are out there in the literature already. So that was mitochondrial, X chromosomal, and autosomal. I made sure to adjust mitochondrial diversity for climate, minimum temperature, because in this study by Francois Ballou and colleagues, there was actually an association between mitochondrial diversity and climate. So I made that adjustment for climate to get, I guess, a truer, a better representation, a better recovery of the out of Africa, of the across Africa expansion signal. I used various tests, correlational mainly. I did some analysis with linear regression, but I won't be mentioning that because of time. And for each diversity, I found the best candidates for being the origin. These were locations from which there's the strongest decline in diversity as we move away from the origin. And I used the Bayesian information criterion to go about that. Now, to estimate where the origin is indicated to be across variables, I came up with a simple method a few years back, and I used it again, and that's to rank how good each location is at being the origin based on correlation coefficients. So doing that ranking within each variable, and then taking an average of how good each location is across variables, across diversities. On to the results. So when it came to the cranium, we had a bit of a problem. So first of all, I used 25 dimensions, and we can see here there's a bit of a difference, a bit of a separation between the data sets. Well, it's previously been noticed that when it comes to the Hanihara and the Howell's data sets, there seem to have been differences in how cranial landmarks were defined. So even though dimensions may be labeled the same, they might not actually be measuring the same thing. So has been previously noted that 11 dimensions seem to be the same. So when I did my principal coordinates analysis using RMET on those 11 dimensions, didn't seem to be a problem, no separation between data sets. So I carried on my analyses using those 11 dimensions only. For each diversity, I actually found a significant decline in diversity with distance from somewhere in Africa. I use examples here of the cranial shape diversity and mitochondrial diversity. And it's also worth pointing out that the declines, the correlation coefficients were strong if we use Cohen's benchmarks. So they were all minus 0.5 or greater or lower, depending how you think about it. So here I show maps of Africa, one regarding each type of diversity. In each map, the darker grey area indicates the best candidates for being the origin of the across Africa expansion. Each of the darker areas covers various parts of Africa. There's variation, but at least each one covers some of Southern Africa. When it came to mitochondrial diversity, we can see that the area is noticeably larger, but for mitochondrial diversity, the dataset I used didn't include any Southern African populations, so that probably explains why it's so much larger than the others. For X chromosomal diversity, I probably could have adjusted for climate. So whilst the previous study, Ballou and colleagues did not find an association between X chromosomal diversity and climate when diversity was adjusted for distance from Africa, I'd forgotten that a few years ago in a preprint, I'd found an association, less diversity with a cooler climate. So if anything, the present study could be underestimating the across Africa signal in X chromosomal diversity. Now onto the good stuff, I guess. Finding out if there's an estimate of the origin across diversities. It can be seen that a Southern African origin was indicated for genetic diversities and was also still indicated when cranial shape diversity was thrown into the mix. Therefore, there seems to be a general indication of a Southern African origin. But as I alluded to earlier, some caveats. Admixture is thought to affect origin estimation. admixture might not be behind these results, however. Why is that? Using linkage disequilibrium, Brennan and colleagues found that linkage disequilibrium indicated a sudden origin. The agreement between their findings with linkage disequilibrium and my findings with diversity in general could be a bit surprising, because admixture, you'd expect that to affect linkage disequilibrium and diversity. You'd expect it to affect their ability to indicate the origin in completely opposite directions. So finding a similarity in suggesting a sudden origin maybe suggests that admixture is not to blame. However, Karina Schlebusch and colleagues did not find that a linkage disequilibrium derived measure suggested any particular origin. So we don't really have a broad indication yet of linkage disequilibrium and its ability to suggest the origin. So got to do a bit more work there. And that future work could actually draw some inspiration from some work on the Bantu expansion, where masking has been used to admixture has been masked out. So that technique could be used in research on the across Africa expansion. Also, some other reasons to be doubtful of Southern African origin, perhaps. There's been some recent research, a preprint that looked into the predecessor ancestry of Eurasians, which may come from which may be actually the African ancestry. So maybe from the African ancestors of the predecessors of Eurasians, if you get what I'm saying. And that ancestry seemed to be lowest in Khoisan. So if the across Africa expansion, if that ancestry does represent the across Africa expansion, having that ancestry be at its lowest in the Khoisan would go against the expansion having begun in the south. Interestingly, if we look at the history of populations, there actually was evidence of a decline in effective population size around the time of the out of Africa expansion. Khoisan were the least affected, so that seems to be consistent with a across Africa expansion originating in the range of Khoisan, which before the Bantu expansion went from the south to the east, so still the south. But it could be climate which caused that, so who knows. And lastly, recent study by Ragsdale et al. Not really much time to go into it, but they looked, I guess, they used more advanced analyses to look into population divergences and fusions, and took gene flow into account. And it made it seem really unlikely that non-Khoisan expanded from Southern Africa. It seems to be a fusion between the Central Western African and Eastern African and ancestral populations, but their definition of these ancestral populations was contributed to by the locations, contributed to by present day populations, so not perfect. So to sum up, there seems to be some evidence of a Southern African origin. Certainly there are trends which are there, and it may seem to on the surface point to a Southern African origin more than an Eastern African origin, but got to look into admixture. This non-African, the non-Africans' predecessor ancestry, if that gene flow into Africans is actually the across Africa expansion, and we've got to look into the lineage of non-Khoisan. And thanks to Professor Andrea Manisa for making available the Manisa et al. Data, which is a subset of the Hanahara data, and these are some references. Thank you. That's the end. Any questions? I guess not.
Cleo, you're muted.
Oh, I was saying that we might have time for a very quick question. If you have a question, please raise your hand and unmute yourself, or use the Q&A box. But I think it's almost time for Khalida's talk. So thank you very much, Zarus. Khalida, you can now share your screen.